My name is Quinton Smith. I'm an assistant professor at the University of California, Irvine in the Department of Chemical and Biomolecular Engineering. I'm really excited to present a new Keystone conference being held May 11th through 14th in Whistler, Canada, on the subject of organoids, engineering innovative approaches for basic and translational insight. And this is really exciting for me because I'm a Keystone Symposia fellow. My vision for organizing this meeting is really capturing the interdisciplinary nature of really advancing our understanding of organoid technologies. As an engineer, we have a lot of great approaches that can be used to control the microenvironment of organoids. And I really want to use this as a forum to highlight the different approaches that are being used to control the architecture, structure, functionality of these organoid systems, and how they can actually be leveraged for studying basic developmental processes as well as disease modeling. So with organoids, we have these self-organized stem cell derivatives that comprise of many different cell types. And coupling our understanding and development of these organoids with single cell biology really gives us unprecedented opportunities to really understand how tissues co-emerge, how these tissues are actually able to differentiate. And I think it's a very special opportunity with this joint session. This field is actually very, very new. The first kind of descriptions of these tissues really began in the early 2020s with human-derived stem cells. There's been a massive body of work understanding development from mice, for example, but the idea of actually taking human material or these human cells and being able to capture some of the features of early human development is completely, unprecedented. And it really speaks to the innovation and how quickly this field is moving. And I really am excited about the ability to capture these advances with advances in single cell biology, where we can actually look at, for example, transcriptomic data of human tissues and using these gastruloid models or these early developmental models to see how closely we can actually map the cell types that are being generated in a dish. I think one of the most important elements that was really going to be captured in this meeting itself is how we can use or leverage these technologies for translational applications. So there's been a re-emphasis of actually using human cell-based models as preclinical test beds. So we're really excited about, while we have these technologies, how can we make the model systems even better? And one thing that engineers appreciate, for example, is the nuance of the extracellular environment in which stem cells or organoids reside. So for example, how can we use synthetic materials to actually mimic human tissues in the body? The value of this conference is very unique in that we have representatives from the biomedical engineering field, which I think is something that's very unique for people that are studying stem cell biology. So I think this will empower many different disciplines to learn about new approaches and trying to solve this paradigm of actually bridging in vitro systems to clinical or translational applications. So I really think this conference is going to highlight engineering approaches, but really be beneficial to a wide array of individuals that are interested in stem cell biology, interested in material science, interested in, the advent of microphysiological systems or these so-called organ-on-a-chips. So I really think this conference is going to expose people to the highly growing interdisciplinary nature of the stem cell field. I think this Keystone meeting is really quite exciting, not only to really get access to cutting edge research, but also it gives the opportunity for the next generation of scientists to really network with leaders in the field. So I do want to highlight that we have an opportunity for short talks within this meeting. The deadlines for these talks are February 11th, 2025. And also we have the opportunity for posters, which the deadline is April 17th, 2025. And this really gives, new trainees the ability to showcase their work to leaders in the field. In addition to having very engaging scientific sessions, as we have a joint meeting with single cell biology, we also have the opportunity for a career roundtable discussion where trainees can actually talk about what does it look like to be a professor in academia, or what does it look like to do research in an industrial setting. So these are very unique aspects of the Keystone meeting that I think is highly beneficial to the next generation of scientists. And even for me as an assistant professor, having access to these discussions, having access to leaders in the field is really quite powerful in forging new collaborations. So I'm really excited about this conference being held May 11th through 14th in Whistler, Canada.